thank you all for joining us in this exciting week. This is the sixth lecture, sponsored by and in conjunction with the Center for Evolution Medicine and Public Health. We're grateful to our friends at CHESC who organized many of them, Kathy and Lenora and Jackie. We're talking about you, Kathy. Kathy, Lenora, and Jackie have done amazing things to make it possible to have 6 lectures in 1 week and make this all work. Today's lecture is special. We tried very hard to find a way to get Paul and regular Schmid-Hempel here, and that's because they've been so central to bridging several different areas that are so important. I mean, first of all, they've been very essential in Europe in trying to connect people who do work on evolution and medicine, thanks in large part to their work of encouraging the Wissenschaftskolleg to invite people there, and much of the work that you see happening now would never happen without that effort. Also, their work connects insect biology, a great strength, of course, with ASU, with work on disease and social systems and medicine, and it just couldn't be a more natural thing. So we decided to invite them not just for one lecture, but for almost a 2-week stay to hang around, frankly in hopes that we could find a way of encouraging them to spend more time with us in the future, in whatever way might possibly work. If you'd like to find time with them, please call Kathy, and she can help you see if there's any time. They'll be here for a few minutes while several of us are away. Paul's education began in Zurich, and he continues in Zurich, although he's been all over the world since for his education. As a life, he trained with John Krebs at Oxford. And again, I'm not going to go into all of your honors and awards, but he's gotten what looks like the highest award in Swiss science. He's been instrumental in creating the European Society for Evolutionary Biology and leading that group. And what I like most is his book on evolutionary parasitology. If you want a really good read about solid science, pick up the book. It's just gorgeous in its simplicity, elegance, and comprehensiveness. Today's topic, parasites, hosts, and the crux of diversity.
Thanks a lot. Yeah, thanks a lot, Randy, for these nice words. Can everybody hear me in the background? Is that okay? Wonderful. I need to wear that for the video, apparently. Well, I know we have a busy week, so today's talk has 2 consolations. One is you can always resort to your food instead of listening to the talk. And secondly, it's not about medicine, it's about real animals. So you see what I mean. Okay, the theme is, as you see here, parasites, host, and the crux of diversity. And of course, the first thing you can ask, why parasites and diversity? And give you one example, which is from agriculture. This is farmers using barley, planting barley of particular varieties. These barley are picked up by fungus, rust fungus, that of course reduces the yield. And hence the farmers respond by planting other varieties. What you can see here is actually a time series, so these are points in time, 1967, 68, and so on, where the frequency of the host genotype is plotted, so this is a particular variant of the barley, and against this y-axis, which is the index of infections. And as you can see, as they increase a certain variety here, frequency, so 67, 68, 69, at some point the parasite comes in, it finds its target as it gets more and more common, starts to infect it, so the infection index goes up. Now the farmers respond by actually reducing the percentage of this variety because the yield is going down. And hence, as a consequence, after a delay, the infections go again back down in frequency, and the variety that was used to be heavily infected now gets less infected and can be used again, so it's increasing in the share and so on. So host parasite systems in general have a tendency of leading the cycles. That's also what predator-prey systems do, by the way. But as a speciality in a way, they have the tendency to keep systems variable, both on the host side and on the parasite side. And it's this kind of variability that we are interested in. Now, our system is a trypanosome that infects a host, which is a social host, a social bee, a bumblebee. It's the most common Central European bumblebee. And these are social animals in the sense that the queens come out in spring, found their own colonies, and then afterwards the thing becomes social, so workers are added, and actually the colony grows until it reproduces in late summer or summer, where everything dies except the mated young queens that go into the soil and overwinter the next year. Now we know that this parasite reduces actually the founding success of the queens quite considerably, up to almost a half. So it's a serious parasite if you have it. And actually, it is very common. If you go out in summer and collect the workers, then maybe anything between 30 and 80% of all workers are infected. Now, why bees? Of course, I couldn't resist to put this in. You find this on the internet. You could choose, of course, mice and humans for these kind of studies or similar studies. But if you think about that, and it has become a little bit more in focus in the recent years, this is what you really get with bees. And I'm not quite sure whether this rendering is absolutely exact, but it gives you the idea. If you would lose the bees, and we're in danger of losing many of these pollinators at the moment, you would get this one. So it would actually reduce quite the degree of variety of products that we have generally in store. So bees are not only an interesting system to study scientifically, but they also have, of course, a lot of commercial value as shown here. Now, coming back to our system. The parasite, as I said, is a trypanosome. This is briefly to show where it belongs to. This is the phylogenetic tree. These are our Critidia species. There are several known ones, by the way, in the meantime. They're close relatives of Leishmania. So Leishmania, for example, is also a parasite that is of medical importance, so leishmaniosis, of course, in the tropics mostly. It is closely connected to Leptomonas, to some plant parasites. It is a little bit more distant to the big African and South American trypanosomes, but it belongs into this group. So it has actually quite a lot of similarities in many ways with these classical trypanosomes, except, as you can see in the life cycle, it is a directly transmitted parasite, which makes it rather interesting and easy to study. It infects per os, so the animal ingests infective cells. We know just a few are necessary, maybe a handful. It's very infective. It starts to actually reside in the rectum, so in the hindgut, to multiply to large numbers. And then the infective cells, after a few days already, are shed out in the feces and can, by contamination, contact to other workers in the colony, for example, can infect the next ones. In fact, we do know that this kind of profile is quite repeatable here. And the important thing, as I was saying, is that they're extracellular, which means we can keep in culture and clone them. And it is actually something that's directly transmitted. So we ourselves can infect animals at our will. So it's a simple lifestyle, and it has basically Bombus, the genus Bombus, as its host. If you ask how it is transmitted, I was just alluding to that one. It's transmitted by infective cells that are taken up per os, for example, contamination in the nest. So how is it going from one nest to the next? So we know in the meantime, we've also done some experiment, that it's transmitted via the flowers. So if an animal lands on the flower and an infected animal has been there before, then there's a certain chance that you get infected yourself and carry it back home basically to your colony. And you can see that illustrated in this particular rendering here. This is the mean time you have to wait in minutes between subsequent visits to the same individual flower in the field. These are field data, in this case in Switzerland. And this is the prevalence of infection you have in the field, so in the general population. And as you can see, the higher the prevalence here is, actually the lower you have to wait on average until basically the next, or let's say the time separation between subsequent visits is actually lower if the prevalence in the field is higher. We also did direct experiments to show that actually it is transmitted really via the flowers. So this is a whole entire different field of interest, I think, to pollination biologists, but flowers are not only places of reward, they're also places of danger, so you can get infected. In fact, it is possible, as shown here as an example, to reconstruct the transmission matrix in the field. So these are different species. Shown here are 4 species here, characterized by their color patterns, and 2 times in the year, early July, early August, so months apart. And you can see the arrows, basically their strengths and direction, show what is the likelihood of transmission of a particular infection from, let's say, this species here, Bombus lapidarius, to this species, Bombus muscorum, and so on. So it's possible to quantify these transmission matrices in the field, and thus it makes it possible, essentially, to study the parasites' dynamics and transmission characteristics also in the field. But this is basically now the natural history background I wanted to give you before we go into the more details. Now, another element of the natural history is the pattern of infection. Here I come to the first part. First, I'd like to talk about the diversity in the infections. So, and I mean diversity means genotypes of the parasite, which we assess by microsatellite markers. And in the second part, I'd like to talk about how the host defends itself by certain small molecules called the antimicrobial peptides. And in the third part, I will go into the bacterial load of the gut. So what is the pattern in nature? One of the most telling signatures of this system is that it is highly specific in the sense that certain strains of the parasites, so if I say strains, I mean genotypes of the parasite, are able to infect only certain colonies, or families if you like, whereas a certain family or a given family is susceptible only to a number of strains, but not to all. So there's quite a specific matching between the parasite and the host. And here you can see in one example, one rendering, these are 8 colonies characterized, or families characterized by these ellipses here. And the cylinders here are the infections. These are genotypes of the infection. And if you look closely, or can read it at least in the front line here, you see that every single cylinder here has a different number, meaning in this particular set there are 27 different genotypes, and each single family has its own cocktail of genotypes. So this one has just one infection, number 8, 14, 26. This one has, for example, 6 infections, and these 6 infections are characterized by numbers 10, 11, and so on. But it's indeed true, if you go out in the field and pick up an individual and genotyped infection, you will find that in half of the infections, or half of the infected animals, there's more than one genotype, so there's multiple infection. And all of these infections, almost all of them, 95%, are different from one another. So it's a huge diversity of these parasites. And this is actually something which has always caught our eye and which makes it also a little bit difficult to study. But first of all, I'd like to show you where this diversity comes from, how it is generated. So the big question, of course, whether these trypanosomes are clonal or sexual, meaning that they exchange genes, particularly, of course, when they co-infect in the same individual host. Now, this is just a micrograph here showing a cell. This is how it looks like here in the microscope. A cell splitting or fusing, we're not quite sure about that one. But what we can do, of course, is we can do experimental infections. Remember, we can clone them. We can take strains, infect them in a given individual, like this one here, so a yellow strain and a red strain, and then take out all the offspring infections by the feces as they're shed, put them in a microtiter plate, so clone them, put each clone in a microtiter well, and sequence them and check them with microsatellite markers, and check which ones of those are parental types, genotypes, and which ones are new novel genotypes. This is what comes out. Of, in this particular example, over 2,000 clones, only about 2% were new types. However, of the infections, so these 91 individual bees, hosts, that were co-infected experimentally, about 17% produced novel genotypes. Of course, the majority is still parental, but they produced also novel genotypes. And if you do the calculations, actually this amounts to a very large number if you have subsequent generations of that, and show you that in a minute. So most of them are still parental. Some of them are recombinant, meaning they're nice Mendelian recombinants. And some of them, the green ones, 7%, are new types, so new alleles. And from how it's distributed, we have to assume this comes about by sort of slippage, so that the recombination is not quite homologous, but a slight slippage so that you get longer and shorter alleles. But this is the machinery. Co-infection leads to sexual exchange of genes, so to speak, and we see it in the generation of novel genotypes of the parasite. In fact, the strain diversity is generated within a colony cycle. This is what you can do in an experiment. So this is the colony cycle. It starts in spring. It ends somewhere in summer. This is the number of colony workers in this colony. So the queen is alone at first, starts to add workers. To grow in numbers, peak number is probably 80 to 100 individuals, sometimes much more, and then it declines. What we did is we infected the queen with 2 strains experimentally, took out the infection here, took out the infection here, and took out the infection here as the offspring were departing with their infection that they collected from the parental nest. I'm not going to show you all of this data, but just to illustrate in one example what's happening here, which is quite characteristic. What you see here is a 2-dimensional ordering of all the genotypes. So what you see here is basically the collected genotypes that came out of a colony. This is one example, this A colony, that originally was infected by 2 genotypes of the parasite. Now, the two original genotypes in this space here are these two dots here. You can see it's a principal component analysis, separates these genotypes out. And all the cloud around it here is what came out of those two primary infections. So in other words, you put in two primary infections and it explodes in all directions so that you get a lot of new genotypes, parental recombinations, but also novel ones that were not there before. And if you do the numbers, what we do see is that in this experiment, 5 different genotypes were infected pairwise, so this gives 10 combinations. And of those, we get 409 new, actually novel strains generated in this colony cycle. As you can see, the machinery that generates this kind of diversity is incredibly powerful. So we get a lot of diversity within a brief time. We do not yet know, unfortunately, what selects for these particular strains. And one of the technical problems is that we have so much diversity that we cannot trace it by genotype. Every genotype is new, so it's basically an individual fate of each genotype. But we're working on it. So keep in mind, the parasite is enormously diverse in number of genotypes. It does so by recombination, by genetic exchange, or sexual reproduction, if you like. And it generates a lot of novel strains, for example, in a colony. It's not equally distributed. In fact, some colonies are very good at producing new, so some family backgrounds, if you like, to translate that in another language, are very good in actually generating new strains, and others are not so. And you can see this here. This on the x-axis are a number of different colonies with their IDs, and this is the frequency of novel recombinants that were produced. And you can see most of them are not very good at it, but some are extremely good at it. In fact, in this particular experiment, you can see in gray here, which didn't work, the result didn't come up, we thought that if we prime them, so make them more defensive against the parasite, they will produce more variants. But this was not the case. As you can see here, particularly those that were already primed against this parasite did not produce more strains than the other ones. So the immune answer is not a selective agent to generate this diversity, which in a way is a bit puzzling. But I would take it with a grain of salt because this is a first experiment and it's not quite clear what we get there in the end. Enormous diversity, second part. How can the host defend itself against such a diversity? I mean, that's a huge problem. We all know that the vertebrates, or at least the short vertebrates, so us, have the so-called adaptive immune system based on B and T cells, which are able to produce an enormous amount of receptors by somatic recombination. Until very recently, invertebrates, and in insects in particular, have been thought to be very, very primitive and simple in the immune response. We now know this is not true. We have analogous processes or mechanisms in insects as well that can produce an enormous diversity of receptors, even though the magnitude is not nearly as large as in the vertebrates. So we're talking about maybe 100,000 different receptors produced from a particular gene. In this particular case, it's the so-called DSCAM gene that is somatically actually spliced and recombined to produce receptors of large diversity. So in principle, the immune system of insects is also able actually to pick up a lot of these things. And in fact, you can very easily test and measure the immune response. For example, here, by injecting an agent that generates the immune or elicits the immune response, an antigen, very typical, for example, LPS, surface of gram-negative bacteria. You can inject it in the animal. A little bit later, you take out the hemolymph and put it on an agar plate that is coated by bacteria, a very classical approach. And as you can see here, maybe even from the back, these diameters all differ slightly, and you can measure the strength of the response by just with this zone of inhibition assay. So a very simple system that you can do to know how these animals defend themselves against an infection. Now, currently, we're a bit farther than that, because in the meantime, actually just last week, the bombus immune genome has been formally released. So it is now a sequenced animal, which means it's a serious animal, it's a real animal, so to speak. And you can see, maybe dimly here, it doesn't matter that you understand all parts of it, but this is basically representative of the immune pathways, and it's sufficient to tell you that the bumblebees, as well as the honeybees, they're very similar in their immune repertoire, and they have the classical immune pathways, canonical pathways that insects have, like the toll pathway, IMD, JAK-STAT, and so on. At the end of this pathway, so the pathway is always triggered by receptors that see something, for example, a surface of a parasite, and it goes through cascades, which are illustrated here. So these would be the receptors, by the way, or ligands. And actually, there's a whole sequence of events in the cell, and at the end of the whole sequence, you get effector molecules. And I'm looking at these now for the second part of the talk. In insects, these are the antimicrobial peptides. In fact, we also have them. We produce them on the eyes. Otherwise, our eyes would be completely infected all the time. We produce them on many different epithelia. So antimicrobial peptides are widespread among living organisms. Even plants have them. Insects, particularly the bees, have 4 kinds of them: abacin, epidacin, defensin, and hymenoptacin. So these are 4 peptides which are actually characterized. We know the sequence, and we can look what they're doing. The first thing we had been interested in is, given this huge diversity, so looking at these AMPs, antimicrobial peptides, given at this huge diversity of strains, parasite genotypes that attack the host, and we know there's a specific response to these ones, data not shown here, would actually there be variation between these peptides among individuals or families in a population? So we went about, collected a lot of animals, sequenced these particular peptides here, and checked how the sequences would look like. The answer is there is variation and signs of selection among species, so different species of bumblebees, but there's no variation whatsoever that we could see within a species or even within a population. So in other words, at least a simple idea that the huge diversity, genotypic diversity of the parasite would be matched by a large diversity of, let's say, effector molecules, for example, is certainly not there. And in fact, there are not so many signatures of selection. One is very clear here. This is aubergine, which is something that's involved in the antiviral pathway. And the antiviral pathway is known in many organisms, particularly insects, to be highly evolving or rapidly evolving. So it's always a strong selection signature there. But here, we don't find anything within populations. And so we stuck a little bit with the fact that these animals are attacked by a number of parasites, different kinds, genotypes of parasites. They respond differently, kill them differently off, but yet those effector molecules which we think act there are not variable per se. In fact, we can see, and that's the idea now, that these peptides may actually act in synergy. So maybe alone they don't do much, but if you start to combine them, they may actually have an effect. And this is the first test we did, which is interesting against E. coli, so bacteria. And these are our bumblebee species peptides, hymenoptacin, abacin. And you can see as you increase the dose of hymenoptacin, you generally get a decrease in the number of surviving bacteria, measured here as the number of CFUs, so colony-forming units, after the peptides have been added for a certain while. So the more of this peptide you add, the more it kills off the bacteria. Wonderful, so it works. But the effect is actually dependent on the addition, or you can modify this effect by adding a second one, which is abacin here. And as you can see, the more abacin you add, the stronger this killing effect gets. So this is a synergistic effect that these 2 peptides work together. Now, Ben Zett, who did this work, actually is now in the States, in Illinois, and actually we have repeated that with some colleagues at the University of Gießen, and it shows up also in the micrographs, so electron micrographs, and so on and so on. More sophisticated tests. These peptides are rather efficient against these bacteria, E. coli, actually also against other bacteria. And of course, this touches to the idea, which is absolutely not new, that antimicrobial peptides from insects, for example, can form the next generation of antibiotics that we urgently need as our standard antibiotics lose effect over time. So it does work. Now, the idea behind this is if the genes and the peptides that are formed from these genes are not variable per se, but we nevertheless see a highly variable response, maybe the clue to that is that these peptides are used in different ways. And actually, that depending on which strain, which genotype infects a particular host, a cocktail of these strains, of these peptides is produced that is particularly able to hit this particular parasite. So the defense may not be by genetic variation or strict production, but by varying the cocktail of synergistic interacting peptides to kill off these parasites in due manner. That's the general idea. And you can sort of draft it here by saying, okay, this is expression of defensin, this is expression of apidacin, two of these peptides. And maybe a particular colony, when it's infected by strain A, will produce this mixture here. And if it's infected by strain A1, will produce this mixture. And the same could be true, actually, if you do it in colony B, so another colony, this is colony A, another colony, they would differ, for example, by reacting with this cocktail to strain 2 instead of this cocktail, and produce this cocktail instead of the other one as compared to A. And as you can see, it is possible to think that actually, depending on who infects whom, the expression of these antimicrobial peptides is different and matches this kind of interaction that we see phenotypically. Is this true? Well, Seth Baribault, who was in our lab, is now also moved to the States here in Carolina. He did an experiment where he infected a number of colonies in a pilot experiment, so to speak. So these are different families, colony K, L, S, and T, and infected them with different strains, clones. Recall, we can clone them, infect them in a given dose, 68, 75, 161. And then for each combination, he was taking out the expression profile of a number of genes that we knew now from the genomic work would be involved in the defense, in the immune defense pathways. What comes out? I'm of course not showing you everything. These are 6 genes. There were 8 cases, I must say 8 genes, which showed a significant interaction signature. So in other words, which showed a differential expression depending on who infects whom. So this is the interaction term we're interested in. Remember, we tried to explain this interaction, this variation on both sides. Now, interesting enough, our usual suspects came up, like 2 peptides, epidacin, abacin, and some other things that you don't need to read. We actually don't know what they're doing anyway. So there's some serpins, there's some receptors here, like leucine-rich repeats and so on. But if we home in, for example, on this one, you can see probably better what I mean. So this is one of these peptides, and it's across these 4 different colonies. And each panel here has, in different colors, the expression depending on the strain that is infected, so 68, 75, and so on. And what you should see is that over these panels, if you move from colony K, which has such a curve, to colony L, which has such a curve, colony S, which has such a curve, colony T, which has such a curve, and so on, if you then put this into a statistical analysis, and over in this particular case, you'll see that these colonies react differently to, depending on who infects them. So what we do see actually at the transcriptional level indeed is that these peptides are expressed in different quantities or in different strengths, and thus form a different cocktail that is able to actually defend against this parasite. So we do find this on the expression level. There is a signature of this interaction effect, and to our knowledge, this is actually the first one of this kind, to our surprise. Just to add, we know that they're effective against the parasite because a partner lab in Leicester, Eamon Mallon and his colleagues, did a study where they actually knocked down these peptides by RNA interference, right? So they knocked down the effect of this, or the production of these peptides, and as you can see, the infection is higher if abacin is blocked, it's not effective. It's higher if defensin is blocked, and you get no effect in the control, which is Nautilus, which is also an immune component, but which is not involved in this pathway of the antimicrobial peptides. So there's fairly good evidence that actually these peptides are effective against these parasites, and they respond in the expression depending on who infects whom. To put this one step further, a student of ours, Moni Marxo, is currently still finishing up, did a study where she was lucky enough to have collaboration of Andreas Wilczynskas in Gießen and his colleagues that produced these peptides, so our bumblebee peptides, synthetically. One of them, defensin, was terribly difficult. The other ones are rather straightforward. Well, I'm not a biochemist, so I say easy. Maybe it's not that easy after all. But they managed. And what we did in this experiment is to try to combine this in vitro. Again, remember, we can keep in culture. So we add these peptides and see how they kill off the parasite. And what she did is have different combinations. So for example, in a particular combination of a certain amount of defensin, a certain amount of abacin, you get a growth of this parasite in replicates, and you can then do some statistics, some magic here, and for example get a few numbers out which characterize the effect of this peptide combination on the growth of the parasite. For example, the maximum level of cells that is reached in the culture, which I show here, and then ask whether this maximal level depends on the combination of these peptides. And yes, indeed it does. That is shown here. First of all, this is a horrible cloud of points, but essentially that's how the raw data look like. Amount of defensin, amount of abasin, so combination of two peptides, and how much actually you get out, how much the parasite grows. Now, it's a bit more sophisticated than that. If you now analyze it, I show it here in one example. This would be essentially a contour plane. Again, this is the amount of abasin that you add, amount of defensin that you add, and this is now a fitted surface for, in this particular case, the maximal level reached in the infection in this culture. And what you can see here is that there is a minimum point. This is the optimal point for the host. What he wants to do, in other words, is to reduce the maximum infection as much as possible. So in this particular case, this is the optimum combination the animal should do between defensin and abasin in order to reduce the parasite as much as possible. What we can see in sin analysis, because this is statistically new terrain, there seems to be a difference between this, as we said before, between the strains, also difference between the colonies that respond to it. But the fact is that in vitro, by combining these peptides in different ratios, synthetic peptides, we see that we get a synergistic effect just as it's reflected actually in the E. coli case, as it's reflected in the transcriptional response that we see, and as we think it should happen. Now the big question, of course, then would be, do the animals always home in on this optimal point? And that's still a challenge. This is what I call predictive immunology, and this a field which is wide open. We need to have a better handle at predicting the immune response. What should you do to actually most efficiently defend yourself against the parasite? This is one way how we can approach the problem. Antimicrobial peptides. Second part, now the last part. Antimicrobial peptides, small molecules, very efficient, can be used in combination, can attack parasite, even trypanosomes, rather efficiently and kill them out. Could also be of medical importance, obviously, or interest. Not quite the end of the story, because in our work, as we went along, we tried to sequence and get a handle on pathogenic bacteria. Instead, we stumbled across the microbiome, as it's known now. So these are the gut bacteria, that's how I define it here. And a whole new world for us at least opened up, although the microbiome has been known for a long time in many animals. It's only, however, the last about 5 years that the work in insect microbiome has exploded in all directions. I take out exception of the termites, where that has been known for a long time, for digesting cellulose. But the real work really exploded in the last few years. In our case, we figured out in a first study that we have a number of groups of bacteria in there, and there are in particular two groups which are of interest here, the gamma bacteria and the beta bacteria. They're actually the most common ones. Over 90% of all the individuals always have that. And if you then look at that across a number of different species, so this is Hortorum, these are different species here. These, the right-hand 3 panels are actually different locations. But what you see here is that in the darker this matrix is, the more common you find this group of bacteria in a particular animal or individual. And you can see that group 1 and 3, which is the gamma and the proteobacteria, are what we call now the core microbiome. They're always present. And then you add a number of other bacterial groups that are there in different proportions. So wherever we look, we have at least a core biome. And in fact, in the meantime, Hauke Koch, who did this work in our lab, is now in Nancy Moran's lab, has figured out that it is basically this gamma proteobacteria, Guillermella in particular, that seems to play a role in what I'm just show in a minute. In fact, if again you do order this statistically, all these microbiomes, so these are taken from different individuals, different species. This is work done by Koch, Hauke Koch in our lab and later. Then you can see that actually this microbiome starts to sort out statistically according to the host species. So in other words, what we find so far is that the bacterial composition of the microbiota varies somewhat from species to species. So each species seems to have its own kind of composition. And that's shown here. So without going into the details, just to mention that is possible. It's also remarkable, and that sets the bees so far a bit apart from the rest of the insects, as far as we know, that the microbiome is not present or in very, very low numbers or taxa or OUTs, more precisely, in the larvae. It seems that in the honeybees this has been shown, and we have good evidence that is true for the bumblebees as well, that this microbiome is picked up after the animal ecloses from the pupae and becomes the adult animal we know. So here is the experiment we did, and others did as well in a similar way. The larvae is kept, its pupae, you take the pupae out, wait until the animal hatches from the pupae, then put the animal either back here to the nest or keep it isolated. If you keep it isolated, it gets very, very few bacteria. But if you put it back in the nest, it gets the full complement, essentially, of the microbiota. So it seems this is something which is socially transmitted. It is transmitted within the family from parent to offspring, or mother colony to offspring. And this is quite remarkable, as I said, because in most other insects this seems not to be the case. It seems that in the larvae it's present. It also tells us that this microbiota is probably not primarily important for nutrition, right? So because the larvae are the stages that feed a lot and grow. So this is to our advantage, because now what we can do is we keep the animals from the larval stage to the pupae, feed them on a sterile diet, and we get animals that are free of the microbiota, or almost free of the microbiota. These are the controls. We have animals that are free of the microbiota, where we add the feces and the microbiota of their colleagues or nestmates, so they get their normal microbiota of the colony. And then we have added in this particular example some cultivated bacteria. Unfortunately, we could at the time only cultivate one species. So we look at this one, control and feces, and our question was, wonderful, this microbiota, does it have any effect on our sort of hobby, namely to understand the resistance against this parasite? So what you do, of course, you infect these animals with our trypanosome and see what's happening. And what's happening was quite striking. You can see here that this is the intensity of the infection. If they have the normal feces complement microbiota, they're very well protected, or reasonably well protected. If you clear it out afterwards, antibiotics, full infection develops. If they have no microbiota, they also have a full infection. And the one bacteria we cultivated at the time was not effective. Now we can cultivate all of them, but at the time it was not effective. So the microbiota seems to protect against this infection. So there's an additional component which is highly exciting in this context. Recall for last time that our main hobby is a bit to understand the variation, so the interaction between parasite genotypes and host backgrounds of families. The question was, would the microbiota add to this variation, to this interaction effect? And the obvious experiment, obvious but not always so easy to do, is to have a number of donating colonies, meaning we take the microbiota from these animals and put them into recipient colonies or recipient workers from different colonies. So in other words, we do a microbiotal transfer here, so a shit transfer if you like. It's of course filtered. And ask ourselves afterwards, if we infect them with different strains again, do we find an interaction effect? And the answer is yes and no. We do find an effect in the sense that the microbiota is effective, keeping out different strains, or it is infective depending where it comes, but it doesn't have a very clear, always a very clear interaction effect. You can see this here. This is where the microbiota comes from. So let's say from the family 43, and was put into family 343, for example. The darker the color is, the redder, the more infection you have. So in other words, the less defensive it is. And as you can see, from the distance perhaps, there's a sort of a vertical striping in this pattern, without looking at the statistic, which essentially tells us that it is depending on who donates the microbiota. So depending what you get, you're defended. There is also a little bit of horizontal striping, which indicates it also depends on who yourself, I mean your identity as a recipient of the microbiota. But it's interesting to see that where you get the microbiota from it is actually more or less defensive or more or less protecting against this infection. Now, you can go through all these different kinds of panels, and it tells you that there's always a significant effect of the donating colony, so the microbiota matters, what you have. There's sometimes an effect of the recipient, and there's every now and then an effect of the interaction, which we're very interested in. So at the moment we think it is effective, it depends what you have, and sometimes you also get this interaction we would like to look for. One final question. If you put in this microbiota, this is something foreign, right? These are not cells of you. Would the immune system see that? Now, to make this answer a little bit more interesting, we selected family backgrounds that were rather resistant. So from this one, this is done work by a Ph.D. student in our lab at the moment, Katrin, and she had selected a few susceptible colonies. So each point is actually the reaction of a colony to the infection. In this particular case, it is diversity, so how many strains, genotypes survive in the infection, what intensity they reach. At the moment, it's just sufficient to remember that susceptible colonies, so these are these points which refer to single colonies or tests of workers with different colonies, are either susceptible in this classification, so if you're infected, develop a high infection, or resistant, if infected, they keep them out. And we wanted to know if we inject microbiota either into susceptible or resistant colonies, would we find a differential response in the immune genes by looking at the gene expression. So we were selecting a number of genes we know are involved in this kind of defensive pathway and looked at expression. And our prediction was, of course, clear. I mean, what would you predict? I mean, which of the two groups should respond more? Susceptibles or resistant? Who is for resistant? So this is science by democracy. Who is for susceptible? Okay, 2-1. So that's statistically not significant. Well, it was actually the case that the susceptibles reacted more strongly. To be honest, I would have expected the opposite, right? If you're resistant, basically you raise your defense, and as a result, you're resistant. But this was not the case. The resistant ones, this is shown the expression level of basin, epidacin. It goes from low to high, so high is here at the lower end. And you can see the susceptible colonies in both expressions of these two peptides. Remember, these were also the ones that came up in these interaction panels, where we looked at the transcriptional response. In both cases, actually, they responded more to the infection, or to the microbiota, I should say. This is not an infection, it's just a microbiota added than the resistant ones. Now, of course, this is not an infection in the sense that it's defending against the parasite. It's a response against the microbiota that you want. So in this particular context, of course, that would make sense if you basically are more tolerant to the microbiota if it helps you to defend. So you would actually see that the resistant ones would actually step up the response less than the susceptible ones. And they gain because they keep the microbiota. But first of all, if you think about foreign, non-foreign, you should think it's the opposite. Okay, final slide. In the meantime, we're at the stage where we go for deep sequencing for this microbiota with the MiSeq. And you can see at the moment, we're a bit lost to see that if you do MiSeq, there's not much variation. There's a lot of scatter, not much variation in the diversity of OTUs, so taxonomic units we find in the microbiota across the family backgrounds or colonies as we usually take them. So this is the moment where we are and try to make sense of it, but I'm sure we will succeed at some point. So let me summarize, say, this is basically a pollinator host parasite system. It has many aspects which are of interest to a wider range of questions. We know there are specific responses. Host and parasite types, genotypes, backgrounds respond specifically to one another. We know that the parasite has a mind-blowing diversity in genotypes, so that we cannot even track a genotype through the season. That was our initial naive hope, just dissolving every so often. It's generated, this diversity, by genetic exchange, which, by the way, is one of the few examples in trypanosome where this genetic exchange is known to occur. It's the highest rate so far known, actually, in the trypanosomes by far. It is the case that the classic genetics, so the sequences of these AMPs and other components of the immune system, shows very little variation. I call it invariant. It's not quite invariant, but it's disappointingly low. We think that the defense is by variable expression, particularly of these cocktails of antimicrobial peptides, which are very interesting molecules by themselves. And we know that the microbiota is also important. It plays a role there. And of course, since the microbiota sits in the rectum where the parasite sits, and it's actually affected by antimicrobial peptides, there's a link there which we have yet to explore, but we don't know actually what it means. With that, I thank various people that have not been mentioned and have been part of the team, of course. And without money, nothing runs here. And without your attention, of course, nothing sinks in. Thanks.
Any time for questions? I'll let Paul comment.
Okay, yeah, maybe.
Do repeat the question so that
Sure.
In the germ-free mice, the mice are kind of sickly. In germ-free bees, are they sickly or are they?
Yeah, when the germ-free bees are sickly, indeed they are. Actually, it is, I mean, it always, as you know, it always seems very easy, but we had to fiddle around quite a bit to find the right dose and to also so they get rid of the microbiota and still are alive. So we had a lot of mortality. I think Hauke has now moved to Nancy Moran's lab, but also has moved now to Texas. He has managed now to improve that kind of thing. So, but it's still sort of tricky to keep them alive.
For the antimicrobial peptides, the colony numbers of bacteria go down to 0.
Right.
Do you get any resistance in the bacteria to the peptides?
Yeah, that's an interesting question. I'm sure you allude, so when the bacteria get resistance to these peptides, you allude to this discussion whether they are such that the bacteria have a hard time to evolve a resistance against it, right?
Do they evolve resistance to these
Well,
peptides?
I think they do, and we don't know for these ones. We have not done the test because you have to run an experimental evolution experiment, which would be easy to do in principle, but other people have tried to do that, and they find that the resistance actually comes up.
Yeah.
So I think it is a myth that you think there's anything that the bacteria cannot resist to at some stage. It's just more or less difficult, I guess.
Yeah.
Here it's not easy. I must say it's not easy.
But perhaps the combination might be difficult
Yes.
to evolve resistance to.
Exactly. Thanks for this question. Actually, that's probably something I should have pointed out. I think the nice thing behind this combination therapy, so to speak, would be in fact that it is much more difficult for the parasite to get to grips with it, because if it infects colony A, it is hammered by this cocktail. If it infects colony B, it's hammered by this cocktail. And this adds to the problem of the parasite to actually evolve resistant, because it jumps from colony to colony and so on. So we think this is if anything, will be a very clever trick of the host to actually be effective, despite the fact that it has only 4 peptides to its disposal, right? Not hundreds, just 4. But by sort of fiddling around with the combination, it may actually be much more difficult for the parasite to cope with it. Indeed. Yeah.
Does resistance to one peptide make the organism more vulnerable to others?
Good question. I don't know in this particular context. I don't know. I think in the literature, those people that have looked at it generally don't find these kind of things, as far as I know. One part of the explanation, of course, is that some of these peptides, that's something one should know, of course, in the background, is that some of those peptides particularly attack the membrane, one thinks, and has good evidence that this is so. So by opening pores or destroying membrane, such that the other one can go into the cell interior. And if that is the case, you wouldn't expect a particular, let's say, combined resistance in some sense. John?
Just kind of wondering what happens over winter, and is there, you sort of get regional effects where, I guess the queens must have it, right, and start putting it on flowers. And
Exactly.
so does it happen just if you're bad luck, your colony's in a place where you're next to a queen, or do they all have it? Are the trypanosomes in all the flowers?
Yeah, well, that's a good question, and I didn't go into this data. But the system works as you described. I mean, as I was saying in the beginning, at the end of the season, everybody dies except the young daughter queens that have been mated and go into the soil. And that's the only way the parasite can overwinter, that we know, because it cannot persist outside for very long, maybe minutes. But then, of course, in the In the flower, the nectar, it's well protected from UV light. It's in this nice sugar solution, essentially. There it can stay certainly effective for a while. But outside, it cannot. And of course, all the flowers are gone in it, obviously. So that's the only way it goes from one season to the next. And we know that the queens are very effective in defending, which doesn't surprise you after all. So the percentage of infected young queens that come out in spring is actually rather low, so it's probably about 5%. Those that have it have a hard life. They probably lose half of their fitness. So what we think is what is happening, we have some evidence to actually buffer that, is that as soon as the queens come out, the colonies start to grow, it's transmitted horizontally to other colonies like hell, via the flowers. And that goes very, very fast. So if you put out a colony early season, within a week, this colony is infected. So it goes very fast. And then it starts to generate new diversity, and just a tiny, tiny bit of this diversity, parasite genotypes, seems to be able to jump onto new queens and go to the next season. So that's how the system works. It's always a bottleneck, a serious bottleneck for the parasite. It's presumably also why this sex is not a bad idea. You start to diversify as soon as the infection builds up. Yes.
Hi. I thought it was great that you started to explore the effects of the microbiome on the, I guess, susceptibility of the host. And I was wondering, when you started to explore the effects of the fecal transplants on the host, was that between colonies or between species?
No, within colonies, the same species. It's always the same species here from terrestris. That's between colonies.
Okay.
Yeah.
And are you thinking about doing future experiments, doing fecal transplants between different species within the same region?
Yeah, we've been thinking of that. Part of the problem, of course, is we like to raise them in the lab, so you control everything. We can raise a number of species. Not all are as easy as terrestris. That's why it's also commercially used species, like in porcines here in the US. But they can do it for a number of species. We can do it. We do actually continue on a number of experiments here, which are not of the kind you were mentioning. We were rather interested, for example, how this community builds up. Whether we always get the same community in a given host background, for example. That's a very important question, whether this is independent of that, so acquired essentially by the environment, or as we think it's part of it is acquired by social transmission. And we have some evidence that the beta protobacteria, they're acquired mostly by vertical transmission within social context, and the gamma protobacteria seem to be more often acquired by the external sources, so let's say flowers, maybe other bees drifting in your colony or something like that. So it seems to be a distinction between these two things. So it's also along these lines a bit.
One or two more questions? I mean, you heard Seth talk about Nosonia, where the larvae also have basically no microbiome, and then kind of it appears miraculously when they become adults, right? We don't know where it comes from, but it's a filter effect, right? So they come from the outside and they just allow a certain proportion of those. So in your experiments, when you did that, is it really so if you keep them isolated that they don't acquire it, or you have to give them basically antibiotics to keep them sterile?
No, they don't acquire it. We tested that. So
how long does that? So they basically emerge, and then that's different from Seth's example.
Yeah, exactly. That's what I'm saying. It's different because if we leave them as larvae in the colony and we let them pupate, they take out the pupae and take them when they hatch as adults, take them away, keep them isolated. They don't get it. They have maybe one or two bacteria, which is not the ones that are typically there. And if we then take the same animal, or another group, of course, put it back to the nest, they get it. So they get it after closure. And we know that, we have checked that with molecular markers, so we know the profile, and it's just not there. So if they're not in the natural context after hatching, they don't seem to have it.
Which indicates that they either have a really strong already immune system themselves, I mean, during metamorphosis, or there's some way they sterilize the cells, right, so that they don't get that, which is everywhere in the colony, basically.
That's true. That's true. That's the amazing thing, right? That the larvae are fed in this species. I mean, bumblebee species differ a little bit how they raise the brood. In this species, actually, the adults sort of regurgitate things into the brood cell. So it's surprising that they at the same time don't regurgitate the microbiota or somehow transfer it, unless it comes out the other end, right? That you cannot regurgitate it really. In the honeybee, people have done extensive experiments. They don't get it. Here we have not yet screened the larvae per se. We know from secondary evidence it's not there. But it's nevertheless surprising that they don't have it, or why they shouldn't need it, of course, I don't know. I mean, presuming it has nothing to do with nutrition. They have some other few bacteria, apparently, but not our microbiota that the adult has. So it's a nice example of social transmission, right? And if you like a tradition, you get the microbiota from your parents. Yeah.
Are the microbiomes in the bees resistant to the antimicrobial peptides that the bees make?
We don't know yet, but my bet is not. I mean, of course, depends on the dose.
Yeah,
But
yeah.
I think you can kill them all.
Yeah, sure.
Them, yeah.
But more resistant than, for instance,
Than
other
E. coli
bacteria.
or something like
Yeah,
that. Yeah,
yeah.
We don't know yet. So that's, of course, one of the interesting bits. One last question.
So on one of the earlier slides, you were showing that some of the colonies had multiple parasite infections and others only had one.
Yeah.
So if there's any evidence that some of the parasites intentionally reduce the susceptibility to new parasites coming in, or do they more or less play well with one another?
Yes. Actually, I didn't show that, but we have from the transcriptional data, we have good reasons to believe that these parasites are fiddling around with the immune response, because our best infectors are the ones that actually release the lowest response. And that's a bit strange in normal terms, unless you think they're good infectors because they're able to reduce the host response. That's what we really do see. Whether this actually leads to more infection afterwards, we have done some experiments where you can see a kind of facilitation of that sort, but the effect doesn't seem to be particularly strong. But this manipulative element, we're quite sure is there. Absolutely. And that's not unusual for all parasites do some kind of manipulation. Okay, one very last one.
So I was wondering about behavior. You know, do they defecate in the colony, or are they always fly out? I mean, is that, I wonder if it's a behavioral response to try to reduce infection, and also maybe to avoid flowers of other colonies.
Yeah, good point. I mean, we know they're defecating outside, but we know that if they're infected, they don't behave differently, and let's say leave the colony more often than if they're not infected. So it doesn't go that far. And in fact, we also know from experiments that don't know if the sister is infected, which you would think, right, that they sort of push them out or harass them more often, which is reportedly true for some honeybee disease. It's not quite clear how good the data is. But in this case, they don't sense it, that they're infected. Okay, thank you very much.